Hello everyone and welcome to another AlphaLyse webinar. We're so glad that you are spending the next 30 minutes with us. My name is Martin Asker Hansen and I'll be your host today. I work as a protein analysis advisor here at AlphaLyse, and with me today I have Thomas Kofoed. He's the CEO and co-founder of AlphaLyse, and he'll present this webinar with the title, Why Do We Need Orthogonal HCP Analysis for Monoclonal Antibodies? Thomas has been working with mass spectrometry and protein analysis for more than 20 years, and he'll go into this presentation in a minute. Also with us, we have our colleague Jette. She'll take care of the technical part. So if you experience any issues with sound or image quality, please let her know in the chat. Today's program will be first with the presentation by Thomas for around 20 minutes, and then afterwards we'll go over to some questions. So if you have any questions, please put it in the chat as well, and we'll pick up on those in the end. But with this, I'd like to pass it on to Thomas and get started with the presentation.
Yes, thank you. I also like to thank you for joining in today. As you can see, the presentation title is a question, and this is a question we get quite often from our clients. Why do we need an orthogonal HCP analysis for monoclonal antibodies? We already have quite pure monoclonal antibody, and we have quite good ELISA for monitoring the host cell proteins. And I think there are some good points here why you should use an orthogonal method. First of all, I mean, no ELISA will have 100% coverage. So to ensure that you don't have any HCPs that are not detected by your ELISA assay, there's a good reason to use an orthogonal method. And then most monoclonal antibodies are given in quite high doses and multiple times. So even host cell proteins at very low level can actually jeopardize the project and causing issues later on. So that's the main reasons for why you should use an orthogonal method for HCP analysis. So why are we here today, and what is it that AlphaLyse has done over the last 20 years? We are a contract research organization specialized using mass spectrometry to help our clients developing new biologics. We have done that for almost 20 years, and for the last 7 years we have been developing this method for looking at host cell proteins using mass spectrometry. We had 10 PhD level scientists working on this. And for the last 4 years, we've been providing this as a service to clients all over the world. And we have now been involved in more than 150 different HCP projects using mass spectrometry, probably making us the most experienced using mass spec for host cell protein analysis. A lot of our clients, once they start using this, they have requested the increased quality level. They have asked us if we could qualify and validate the method, and we are working on that, and hope that within the next year and a half, we will also have the possibility to offer the analysis under GMP. And finally, I want to mention that all the development we have done is done together with clients of ours, so we have a great focus on clients' needs and have developed these methods together with clients. So first, I think I would just take up this, remind us a bit about why do we have to look at process-related impurities, and why is it interesting to look at this. And I have shown here a review made by Martin van der Laan and colleagues a few years ago, where they actually look at, in the literature, they try to find examples where host cell proteins has caused problems and delays in projects. And I will just use some of the information in this review on the coming slides, just to remind us, or remind you about why is it we have to monitor process-related impurities. First of all, there's of course the issue with patient safety. So we need to look at the host cell proteins to make sure that we don't have host cell proteins in our drug substance that can cause immunogenicity and an immune response. And we also want to make sure that we don't have any host cell proteins that can cause biological activities. And there are a number of examples in this paper. So I've just listed them here. I don't want to go through them all, but for example, immune system activators like cytokines has been monitored and caused problems, and also anti-drug antibodies with cross-reactivity against antibodies have been seen in different projects. And finally, also hitchhiker proteins, HCPs, like clusterin and phospholipases has been detected in different projects and caused delays, and even some projects that were failing completely. So, of course, we need to look at the host cell proteins from a patient safety point of view. You also need to look at it because you would like to have a stable product, and some host cell proteins could actually cause degradation, modify the drug substance, and thereby lose of activity. And there are also a number of examples where excipient degradation has taken place. And especially we think about the lipases that can cause degradation of the tween, typically used together with monoclonal antibodies. But also enzymatic activity like catepsin and disulfide isomerase has been found in different projects and causing problems. Finally, you also need to document the manufacturing consistency, show that you have a robust purification process. And again, here you can use, or you need to show the process-related impurities during your purification and show that your process is removing HCPs to a certain level. So that's the 3 reasons why we need to monitor host cell proteins. And I will just go to this next slide where we have listed the reason or the good thing about ELISA, but also some of the not so good thing about ELISA assays. I guess you all use ELISA for monitoring host cell proteins today. It's a very nice tool for high throughput. It's very easy to put in, or easy to put into a QC environment, and you have high sensitivity for the antigens that you have antibodies against. The disadvantage with ELISA is that you don't get any detailed information about specific HCPs. So you just get a number for total HCP, but no detailed information. You also have long development time. If you need to develop your process-specific ELISA, you can spend a year and a half, or even more than that, on developing a process-specific ELISA. And this also gives very low flexibility. So if you do process changes later on, then you have issues that you might get back and qualify the ELISA assay again, or even make it from scratch. So that's the main disadvantages with ELISA. I will start with the positive about using mass spectrometry for host cell proteins, because with mass spec you can get quantification of individual host cell proteins. Also, it's very fast to develop the method within typically weeks. It's very flexible to process changes. So if you do changes or adding proteins or whatever you do, you can easily update the protein database used with the mass spec data, and then you can use the exact same assay. And due to this individual HCP identification, you can do risk assessment. So now you can look at the individual host cell proteins and make sure that none of those can cause you problems. The disadvantage with mass spectrometry, and the reason why it hasn't been used that much in the past, is of course that it doesn't have a high throughput. Normally you might have with one instrument 10, 20 samples a day, so not the same throughput as with an ELISA. It's also highly complex, the analysis. It requires a very detailed and complex sample preparation, and you use very sophisticated, very expensive instrumentation, and it is therefore difficult to put it into a QC environment. But the 7 years we spent developing this method has actually focused on these disadvantages with mass spectrometry, and we think we have solved most of them, and at least limited them, and I will just show you why and how we have done this. So first of all, we have automated the sample preparation. So as you might know, the sample preparation before mass spectrometry includes a digest of the sample and the host cell proteins. Then it's purification step before we load them onto the mass spec instrument. We also include standards in all the samples, so we have internal standards in all the samples we analyze, and thereby we can actually control a lot of the complexity and variability in the assay. Then when we analyze it by mass spectrometry, we have established a very robust mass spec analysis, so we don't use nanoflow systems, we use microflow systems, and we also record the data in data-independent acquisition mode, which makes the data more robust and reproducible. And finally, we have automated the workflow for data analysis. Based on these 150 different projects, we have optimized the workflow for data analysis and reporting. So in total, we have control on all these disadvantages with mass spectrometry and have solved a lot of the issues related to mass spectrometry and use it for HCP analysis. So for monoclonal antibodies, we actually have a two-way sample preparation. So we use our standard, we call it quantitative digest method. And here you can see the antibody with some host cell proteins. Typically, before you do a digest, you will do a denaturation. So you would use DTT and an alkylation reagent, and then you would do trypsin digest afterwards. By doing this, you will digest all the proteins in the sample, also the antibody itself. So you will generate a lot of peptides, of course, from the antibody itself, and then the minor peptides will be from the host cell proteins. The second way of doing this is, we call it native digest. It's a digest where we don't do the reduction and alkylation. So we do the trypsin digestion directly on the sample, and by doing that, we mainly digest the host cell proteins and not so much the antibody itself. Then we can use a lot more material, and we can then remove most of the antibodies before we load it onto the mass spec instrument, thereby increasing the sensitivity. The disadvantage is, of course, that some host cell proteins might behave more or less like the antibody, being less accessible from the trypsin, and thereby you won't generate as many peptides from those host cell proteins that have those properties. So we say that the quantitative method is quantitative down to 10 to 25 ppm, and the native digest, we have a lower limit of detection down to 0.1 ppm. So remember, we can't quantify down to 0.1 ppm, but we can identify the host cell proteins. I just want to show you one slide here. This is actually, remember, we have these standards in all the samples we analyze, and we know exactly how much we have spiked into your sample. And we can then see the signal, and here you see the signal from 7 different standards, and you can see they have different slopes, but they are not that different. And that's why this quantification method is quantitative. But if you look at the native digest method, here we spiked in 10 ppm of these 7 standards, and now you can see that the slopes are spreading out more, so it is less quantitative. But what we have seen is that we can still use the data from each specific HCP and compare it from batch to batch. So the precision is still good, it's just the accuracy that is not so good for quantification. So when I'm recording in the following slides or showing data, then with the quantitative method, it's in ppm, and with the native digest, it's number of peptides we have detected. This is a quite complex slide, but it actually shows exactly the way kind of data we generate with mass spectrometry. And as you can see here, we have analyzed, in this case, 4 different samples from a monoclonal antibody project. So we have a sample that contains a harvest sample, so they contain a lot of host cell proteins. Then we have a sample after protein A eluate, and then we have a polishing step, and finally the drug substance. As you can see, we can identify individual host cell proteins in these samples, and you will be getting results both related to the UniProt number, the accession number, the protein name, and also the PI and the molecular weight of individual host cell proteins. And you can easily compare because you have the individual amount in each of these samples. So you can compare for batch to batch, and you can compare between purification steps, and thereby show exactly how you purify your drug substance. So the quantitative method is in ppm, and the native is in number of peptides. We also provide a total number of HCPs using this method, and you can see here it's similar to what the kind of results you would get with an ELISA. So you have the total amount in ppm recorded, and will be able to compare from batch to batch. And just see here that we actually did find some host cell proteins using both the quantitative and the native digest, but none of those were, for the two last samples, were above the lower limit of quantification. So we use this method to analyze host cell proteins in monoclonal antibodies that are already on the market. We had a collaboration with Evidentic, a German company, and they could provide us the aliquots of the drugs that are on the market. And we got these 16 different monoclonal antibodies. So there's different drugs, and there are also actually 4 batches of the same originator drug and 3 biosimilar for the same drug. So we both compare between different drugs and also in between, or looking batch to batch variation. And then we applied these 2 methods that I just shown you, the quantitative and the native digest. When we look at the quantitative data, remember that we only record data above 10 to 25 ppm. We find HCPs in 3 of the samples. And when we then use the native digest, we identify a larger number of HCPs, actually 55 in total in all these samples. And remember that the infliximab is the same batch, so actually it's only 2 out of the monoclonal antibodies analyzed in this study that doesn't contain any HCPs at the native digest detection limit. And here we have the biosimilar, and here we have actually Humira. And I think one of the reasons why it's so much more dirty than the other is actually that Humira originally was not purified with protein A. So that's the reason why we have this high level of HCPs. So this might not look as a problem, but still, if you look into the detail about these host cell proteins, we have here listed all the host cell proteins found in these different samples. One thing you can see is that actually some of the host cell proteins are detected in multiple projects. And there we also, when we look into the list of specific host cell proteins, we can see that some of them are actually of concern. First of all, we have clustering found in this one sample, and this is immunogenic. We have seen this previously. So not so good that we actually detect 7 peptides in this one drug. We also see protein A, and this is typically bleeding from the column. And this is also something you should investigate when you look at your samples, that you should show to the regulatory authorities that you don't have protein A bleeding from your column. And we have one more down here, cathepsin, which is actually an enzyme that can degrade the drug product itself. And actually, we have seen with Humira that the stability of that is lower than if you compare with biosimilars that doesn't contain cathepsin. So it is for sure a problem that you have this one HCP, even at low level, in your drug substance. So the summary of the study is in 50% of the commercial monoclonal antibodies we analyzed, we could see HCPs above 1 to 5 ppm. In 80% of the commercial monoclonal antibodies, we had HCPs detected above this 0.1, 0.25 ppm. The HCP profile vary from drug substance to drug substance, but there are some shared host cell proteins. And we also find that some of these commercially available monoclonal antibodies contains host cell proteins of concern. And I think that answers then the question why we need orthogonal HCP analysis for monoclonal antibodies, because you want to ensure that the ELISA is not missing out abundant HCPs. So that's one reason. So to just be absolutely sure that you don't have HCPs in your drug substance that can cause you problems. You also like to identify potential problematic HCPs early on, so you can actually go back and do process development and thereby remove or limit the amount of these specific HCPs. And you can do detailed risk assessment of specific host cell proteins to ensure patient safety. So I think these two methods, they go very well hand in hand, ELISA and mass spectrometry. They complement each other, and what we hear is that an increasing number of projects actually include both data from ELISA and from mass spectrometry. And with that, I will turn over to the question session. So,
yes, I'll jump in again. Thank you so much, Thomas, for a really interesting presentation. So I'm excited to see what kind of questions we have from the audience today. Yes, so we have the first question here. How low should you measure HCPs in monoclonal antibodies?
Yeah, I mean, as I started saying, monoclonal antibodies are given in quite high dose, so you should measure them low, I think. And we also feel that an increasing number of our clients actually focus on specific host cell proteins, even below 1 ppm, and they have interest in being at least able to monitor specific HCPs way below 1 ppm. So I think once we get more and more knowledge about the HCPs using mass spectrometry, there will be an increasing list of HCPs that we will look for and perhaps detect, way below 1 ppm, just to make sure that they don't cause any problems.
Yeah. So you would say that the trend right now is that companies producing monoclonal antibodies want to go lower, so they...
Yes, for specific HCPs of concern. Yeah,
and then please put in your questions if you have any right now. So I'm thinking we could also talk about the regulatory authorities.
Yes.
What do they say about monoclonal antibodies and HCPs?
In general, about HCPs and use of mass spectrometry, we just attended the BEPA conference about HCP analysis, and there you had representatives from both EMA and FDA. And EMA is saying that an increasing number of the projects they see include mass spec data. And they also argue that the more they see of these, the more they don't require it yet, but eventually they will think that by adding mass spec data, you do a better job than without adding mass spec data. So of course they don't give you a limit or ask you to show to a certain because it's project dependent, but it is an increasing requirement, not a requirement, but an increasing request to add mass spec data.
Yeah, thank you, Thomas. Yeah, and while we're waiting for a few more questions, I think we have a bit more time. I would also like to mention that we have some other webinars on our website with other topics as well. We both go into gene therapy, the method itself. Thomas also mentioned that, we have this method webinar where you can learn a bit more about the specific method we have set up, the details of that. And and also another monoclonal antibody webinar with our colleague Rikke, where she goes through this study of commercial mAbs in even more detail than we did right here. Yes.
No questions? No. So I think we should just...
Yeah, I think we'll just keep it short and sweet today instead of going up to the 30 minutes. So if you have any questions after this, please feel free to reach out to Thomas or directly to AlphaLife via our website. But thank you so much for joining today and listening in. Have a nice day.
You too. Bye-bye.